Hello everyone, welcome to the class. In today's lecture, we are going to talk about C3 cycle. This was proposed by Kelvin and his co-workers. That's why it is known as Kelvin cycle also. Fine. So this C3 cycle is present in all the photosynthetic plants. It doesn't matter they follow C3 pathway or C4 pathway. So here we are going to talk about the steps involved in making food, that are the carbohydrates. So let's see how the glucose is formed in the plant by this cycle. C3 cycle is going through 3 steps. One is carboxylation, that is addition of CO2. Then reduction of that product which is formed at very first. This is PGA. Then regeneration of RUBP. RUBP is the substrate to which that CO2 is going to bind or which is going to accept the CO2 at very first. So these are the 3 main steps. We'll see how these steps undergo. The very first we need RUBP. This RUBP is the primary CO2 acceptor. It is going to accept the carbon dioxide from atmosphere. This stands for ribulose 1,5-bisphosphate. This is a 5 carbon containing compound. This RUBP accepts the CO2 and it is converted into the first stable product, that is 3 carbon containing 3-phosphoglycerate. So this is going to make 2 molecule of phosphoglycerate or phosphoglyceric acid. As this 3 carbon compound is the first stable product, that's why this is C3 cycle. So this is the first step, carboxylation, addition of the CO2 to the substrate in the presence of enzyme called Rubisco. Rubisco stands for ribulose bisphosphate carboxylase and oxygenase enzyme, which have the dual property. We'll discuss that later on. So after adding the CO2 to the substrate, we got the first stable product 3-phosphoglycerate. This is 3 carbon containing compound, 2 molecules we have got. 2 molecules. Fine. Now this phosphoglycerate is phosphorylated by using ATP. So 2 molecules, in that way we need 2 ATP molecules and 2 ADPs are formed as a result of it. So 2 phosphate are added to 2 molecules of this, it become 1,3-bisphosphoglyceric acid or bisphosphoglycerate, 2 molecules, okay. 2 molecules are formed as there are 2 molecules. Now, second step is reduction. Reduction of this first stable product, this one. So this is going to reduced by using NADPH. That NADPH get oxidized to form NADP plus H positive ions and reduce this into 1,3-bisphosphoglyceraldehyde. Whenever acids are reduced, they form aldehydes. So this is an acid, this is aldehyde, okay. So in monosaccharides we have read ketoses, aldoses type of sugars. So here the sugars which are formed, these are like containing aldehyde group in them. So this product which is formed now after reduction is 3-phosphoglyceraldehyde, because from this 2 inorganic phosphates are removed to get the 3-phosphoglyceraldehyde molecule. This will going to follow another reaction series in which different sugars are formed, trioses, pentoses, hexoses, got it, to form the glucose and stored in the form of the starch. So this will continue with different kind of reaction series. Now 2 molecules of this phosphoglyceraldehyde we have got. From this, after undergoing many reactions, we are going to generate this RUBP back. This is required to generate so that this cycle can be run again and again continuously. So we can see that this 3-phosphoglyceraldehyde 2 molecules total contains 6 carbon and this is 5 carbon molecule. So out of this 6, 5 carbon are used to regenerate this RUBP and 1 carbon is going to form the 1/6 of the glucose. And we know that glucose is having 6 carbon. So to make those 6 carbon in the glucose, this cycle is to be run 6 times. Okay, this is only 1 molecule of CO2. So 6 molecule of CO2s are required to form 1 molecule of glucose. So this cycle will be run 6 times to form the glucose. Got it? Now, while regenerating, 1 ATP is used over here to regenerate this RUBP and the cycle will continue. Over here, if we are going to calculate the number of ATP used and NADP used, so we can calculate it very easily. As you can see, the 2 ATPs are used over here, 1 ATP is used here, total 3 ATP in one cycle. And to form the glucose, the cycle is to be run 6 times. So 3 into 6, it means how many ATPs we have used? 18 ATP. Okay. And how many NADP? In one cycle, we are using 2 NADPH. So as it is going to be run 6 times, 12 NADPH. So total consumption of this ATP and NADP is 18 ATP and 12 NADPH to make 1 glucose molecule. Got it? Thank you.